Alright, a very good afternoon to all. I'm Gariel, the Regional Sales Manager for MP Biomedicals for Southeast Asia, Taiwan and Hong Kong. I'd like to thank all of you for taking time off to attend MP Biomedicals' first live webinar in APAC. Today's topic will be on optimizing your microbiome research using gold standard methods. And today's speaker is Dr. Wong Ming, our research scientist for molecular and cellular biology based in MP Biomedicals, Singapore. So without further ado, Dr. Wong, please.
Hi, good afternoon, everyone. Welcome to today's webinar. My name is Ming. So without further ado, let's go to the content of this webinar. So today's presentation will cover on two major topics. Firstly is the microbiome research, where I will talk about what is microbiome and why is it important to study it and how to study it. Secondly, we will look at how MP-Bio can help in your microbiome research by acting as a one-stop solution for sample preparation and DNA extractions. I will also introduce two microbiome extraction kits that are recently launched. And lastly, we will have a summary and Q&A session. Before we start, let's get to know what is microbiome. The term is referring to all genetic materials of microorganisms found in an environment, which include air, soil, water, skin, feces, and many more. The study of the genetic material can provide useful information about the microbial composition of the environment. Here is an example of microbiome study which target on permafrost. Permafrost is any ground that remains completely frozen for more than 2 years, and some of them are thousands of years old. A study has been done to characterize the microbial diversity from permafrost of different geological edge in Alaska. As you can see from figure 1, the edge of permafrost increased with the depth of permafrost tunnel. From the tunnel, scientists have collected permafrost of 19, 27, and 33,000 years old and extract DNA from them. Phylogenetic analysis of the extracted DNA shown that the microbial composition are very different at different geological times, as shown in figure 2. By studying microbiome, scientists can characterize the microbial structure of an environment so to monitor the changes upon events such as pollution and natural disaster. The structure of microbiome can also be used for comparative study. For example, the difference between the microbial community in the feces of a healthy and sick individual. Moreover, the study of genetic material can lead to the identification of new species or new gene that encodes for novel protein or other useful metabolites such as antibiotics. This represents a vast area to explore on because there are so many places on Earth that is yet to be discovered, and each of them as a microbiome. As such, the microbiome study has wide application into different fields, which include agriculture, biofuel, biotechnology, environment, ecology, and healthcare. Take agriculture as an example. The study of microbial population of a fertile soil and their gene expression enables scientists to understand more on the nutrient cycling and nitrogen fixings, and hence provide ideas on how to remediate a nutrient-poor soil for agricultural purpose. Moving on, let's look at the study of microbiome, which is consisted of 3 major stages. It started with sample collection and preparation, and then moved towards DNA extraction, and lastly DNA analysis. We will look at this process in detail in the following slides. The first step of microbiome study is sample preparation. This step is to lyse the cell and release its content, which include DNA, RNA, proteins, and other intracellular substances. There are a few points to be considered during this process. Firstly, the lysis method should effectively lyse all the cells, so the study will provide a biased representation of the microbiome. This is especially true when the lysis method is weak and unable to lyse gram-negative bacteria with tougher cell wall and hence underrepresent them in a composition. Secondly, the lysis temperature. This is dependent on the purpose of extraction. Low temperature lysis method should be used if the target is heat sensitive. Thirdly, the lysis reagent and consumable must be free from DNAs and RNAs to prevent enzymatic degradation of the target. And lastly, lysis reagent should effectively protect the target from contamination, as certain contaminants will bind irreversibly to the targets, For example, humic acid from the soil will bind to a DNA and making it not usable for other downstream applications. There are many ways to lyse the sample. It can be generally split into two major groups, the non-mechanical methods and mechanical methods. Non-mechanical methods can be further split into three types, which is the physical, that uses heat and sonication, chemical method that use detergents, and biological method that use enzyme to lyse the cell. On the other hand, mechanical methods is using homogenizer to break open the cells. Underneath each method is the pros and cons. There is no absolute method for sample lysis, but generally, homogenizer is the widely accepted method for microbiome study because it provides sufficient strength to lyse the difficult environmental sample, and the process is quick and easy. Many of the commercial extraction kits for environmental sample are using this method, including those from MPBio, Kaizen, Omega, Thermo, and Zimo Research. After lysis, we need to separate the DNA from other content of the lysis. DNA can be extracted using conventional method that involve phenochloroform. It exploits the different solubility of DNA in aqueous and organic solvent. So when a polar and non-polar solvent are mixed, they form two immiscible layers. And DNA as polar molecule will partition itself to the aqueous layer and get purified. This method is old and not easy to operate, and it also uses toxic chemical. So in order to overcome these limitations, numerous extraction kits has been developed and available for DNA extractions. A kit usually consists of different buffer and a matrix that can bind to DNA. We will explore on the working principle of extraction kit in following slides. There are different types of extraction kit on the market. They all have DNA binding matrix, which is a substance that can bind to DNA when conditions are favorable. Generally, there are two types of binding matrix, silica and magnetic base. And silica can exist in two forms. The first one is suspension of silica, which has milky color, and hence we name it as glass milk. The second one is a silica compressed into thin layer and fixed within a column, and lysates are passing through the column through centrifugation force, and hence named as spin column. Let's look closer at the structure of silica and magnetic beads. Both of them have negative charge on the surface, and hence under normal condition, DNA with negatively charged backbone will be repelled. However, when a buffer of high salt concentration is added, the positive cation can then form a link bridge between the matrix and DNA, and thus allow DNA binding. Showing here is the general process of DNA extractions, which can be summarized into 5 steps: lyse, purify, bind, wash, and elute. From the left, we have a circle that represents a cell which contains DNA, RNA, proteins, and other intracellular substances. The first step is to break open the cells and release the content. Then, centrifugation can be applied to remove cell debris, and RNAs and other chemicals can be used to remove RNA and proteins. At the third step, the lysate is passed through a matrix which can selectively bind to DNA. Following that, unbound substances are washed off. Finally, pure DNA can be eluted from the matrix. As there are many extraction kits on the market, listed here are a few criteria on how to choose the appropriate one. Firstly, the kit should have wide application. For example, a soil DNA extraction kit should work well with different types of soil, for example, sands, mud, compost, or others. Secondly, an extraction kit can be graded based on its performance, which is defined by the yield, quality, and purity of extracted DNA. The yield should be sufficient for desired downstream applications. High-quality DNA is intact and appears as single band when analyzed using gel electrophoresis, while low-quality DNA will appear to have smearing and RNA contamination, as shown in the image on the right. Purity of DNA can be graded based on absorbance ratio at wavelength 260, 280, and 230 nanometers. A pure DNA should have 260 to 80 ratio at around 1.8 and 260 to 30 ratio above 1. And thirdly, a good extraction kit should be easy to operate and have short processing time. Usually, the standard processing time is around 35 minutes per sample. And lastly, extraction kits comes in different price, and we can define it based on cost per run of a sample. Once the DNA is extracted, it can be subjected to analysis. There are two main approaches, namely the 16S rRNA gene sequencing and shotgun sequencing. The first method uses PCR to amplify the variable region of 16S rRNA gene, followed by sequencing of PCR product. The sequence can be matched with database to elucidate the bacterial composition of an environmental sample. As this method is targeting the 16S rRNA, it is only applicable for bacterial study and is dependent on existing database. It has the advantage of low cost and require only little amount of DNA. The second method aims to sequence all the DNA in sample. This often leads to the discovery of new gene or species that is not previously recorded in the database. And moreover, the analysis is not limited to bacteria only, as the genes for archaea and eukaryotes can also be sequenced. However, the drawback is the high cost and it requires high amount of DNA. So both methods are quite useful for microbiome study, and it is dependent of the research objective of the project. So moving on, I would like to present the role of MPBio products in microbiome research. Let's begin with the upstream, whereby sample preparation is carried out. As mentioned, mechanical lysis is the most appropriate and widely accepted method for environmental sample lysis, which can be achieved by homogenizer. In MPBio, we offer different models of homogenizer, each supported by a range of adapters, and we also have different lysing metrics to target specific sample. They are all under the FastPrep system. This system has been established for more than 20 years, and we are constantly improving our product for wider application and better user experience. These homogenizers are powerful tools to provide sample lysis within 20 to 40 seconds for any sample. Showing over here are the three main FastPrep instruments. The first one is a hand-carry super FastPrep 2, which is suitable for field work. The second one is FastPrep 96, which meant for high throughput work, as it can simultaneously lyse 192 samples. And the third one is the FastPrep 24 5G, which is our latest and top-selling model. It has program optimized for 72 types of sample, or we can adjust the parameter manually. And it comes with a touchscreen for better control, and moreover, it has different adapter for specific needs. The default adapter is the Quick Prep adapter, which can handle 24 tubes of 2 ml sample. Alternatively, we have High Prep and Tall Prep for higher throughput or larger volume. And Thin Prep and Big Prep is meant for large volume handling at 15 and 50 ml. This is the cryogenic adapter, which allows homogenization to be carried out at low temperature, and it is very easy to use. Just add the dry ice into the chamber, and it can prevent heat degradation of sample. And this is the metal adapter, which is suitable for biohazard sample handling, as the adapter is autoclavable. Other than the adapter, there are 16 types of lysing metrics for selections. Each of them has a different combination of grinding material to target specific sample. For example, we recommend lysing matrix E for soil, feces, and other environmental sample. It consisted of 3 types of bits to effectively break open the soil particle and lyse the microbe inside. The common size of lysing matrix tube is 2 ml, but larger volume are also available. These tubes are sealed with O-ring to avoid leaking and cross-contamination, and they are manufactured under GMP and certified to be DNS and RNase free. Moreover, if you already have homogenizer from other company, you can also use these lysing metrics because they are meant to be compatible with other homogenizer. Following lysis, MPBio has offered different kits for DNA extractions. We have kits that are based on glass milk, spin column, and magnetic beads methods. They are named as FastDNA, GeneClean, and MPure accordingly. Each product line is serving on different purposes. The first product range, FastDNA, is using the glass milk technology. There is FastDNA spin kit, which is used for plant, animal, and bacterial sample. FastDNA spin kit for soy and FastDNA spin kit for feces are used for soy and feces sample, respectively. These products are well known for their extraordinary yield advantage due to the glass milk technology, and many literature has reported its high yield and its capability to recover DNA from very difficult samples, even when other competitor kit is unable to do so. The drawback is that glass milk is not that easy to operate, as they require the user to resuspend the matrix and pellet it down by centrifugation for every transfer process. The second product range, GeneClean, has kit that uses glass milk and spin column technology. They are mainly used for PCR product purifications. The third product range, MPure, are fully automated magnetic beads extraction kits. The extraction process is all pre-programmed and done by automated instrument and hence a closed system. As introduced, MPBio has a complete range of products to support microbiome study from sample preparation to DNA extraction. With more than 20 years of experience, we have earned our reputation as the gold standard for microbiome research, and particularly our top-selling kit, FastDNA Spin Kit for Soy, has accumulated more than 7,000 citations, and it has been extensively tested and demonstrated its ability to extract DNA from all kinds of environmental samples, which include activated sludge, contaminated soil, intestinal content, feces, and many more. Showing here are the results adapted from one of the few studies that demonstrate the yield advantage of the kit. It has consistently shown to extract the highest yield of DNA from different samples, and this is particularly useful when the researcher has limited starting material or the sample has very low biomass content. Despite that, we do not stop moving forward. Over the years, we have received the end-user inquiry on the availability of DNA extraction kits based on spin column method and magnetic beads extraction kit with open system. Therefore, our R&D team has worked on a request and present to you the SPINeasy and MagBeads product. SPINeasy is a DNA extraction kit based on spin column technology. It is silica-based extraction kit that is easier to operate as compared to glass milk. SPINeasy DNA kit for soy is available now and more are coming, which include kits for feces, plasmid DNA, plant and culture tissue. On the other hand, MagBeads is the DNA kit based on the magnetic beads method with open system, which means the end user can now troubleshoot or modify the protocol depending on sample types. The kit for feces is available now, and upcoming we will have kit for soy and plant sample. I will further introduce these two products in the next section. The first new product is SPINeasy DNA kit for soy. These are the components of the kit. Currently, we are offering two packaging sizes, and one is the 50 preps and another is the 5 preps trial kit. To extract DNA using this kit is simple. It follows the 5 processes: lyse, purify, bind, wash, and elute. Firstly, lysis is done by using lysing matrix E, which is specially made for crushing soil sample and a microorganism within. Lysis buffer S1 and S2 is used to provide a buffer environment for released DNA, and RNase A is added to remove RNA. Secondly, inhibitor remover S is used to remove proteins and other contaminants by forming precipitate that can be removed by centrifugation. Thirdly, binding buffer S is used to promote the binding of DNA towards colon S1. Following that, wash buffer is used to remove unbound substance, and lastly, DS is used to elute pure DNA from the colon. This kit has been evaluated for its performance using different types of soil, as shown in Table 1. Soil of different characteristics are tested. These include organic soil and paddy soil of high content of humic substance. These are yellow-colored compounds that can irreversibly bind to DNA and thus inhibit downstream applications. Saline soil of high salt content and desert soil of low biomass were also tested. In Table 2, it summarizes the performance of this kit. It is shown that the extracted DNA has satisfying yield and the DNA is intact by showing single band at Figure 1. There is no smearing and no RNA contaminations. The extracted DNA also has very high purity based on the adsorbance ratio, which is near to 1.8 for 260 to 80 and larger than 1 for 260 to 30. The high purity suggested that the DNA is free from contaminants or inhibitors and hence can be used directly for downstream applications such as PCR for 16S rRNA gene, as shown in Figure 2. We have compared the yield performance of this kit with our top sale FastDNA spin kit for soy and kit from Omega and Kaizen. They are used to extract DNA from 5 different soil. And the result shown that both SPINeasy and FastDNA kit from MagBeads has higher yield as compared to competitor kits. And particularly, FastDNA spin kit for soy of Gasmic technology is having the yield advantage by extracting the highest yield of DNA even from the low biomass soil such as the desert soil. In terms of purity, SPINeasy has demonstrated its outstanding performance by having the 260-80 closest to 1.8 and the highest 260-30 value. To summarize, this kit has shown its ability to extract DNA from different soil types, and hence having wide applications. It also has high performance by able to extract high quality of DNA of high yield and purity. Moreover, it is an extraction kit that is based on spin column technology, which is easy to operate and time saving. Moving on, let's look at the second new product, which is the MagBeads FastDNA kit for Feces. These are the components of the kit, and similarly, we will have the 50 prep packaging size and 5 prep trial kits. And we are also having the magnetic racks of 8 and 24 holes to support this kit. To extract feces DNA using this kit, firstly, pre-wash the sample to remove large portion of soluble contaminants. And then the sample is lysed by using lysing matrix E, and lysis buffer F1 and F2 is used to provide a buffer environment for released DNA, and RNase A is added to remove RNA. After that, PPS, which stands for protein precipitation solution, is added to precipitate out proteins and other contaminants that can be removed by centrifugations. Following that, binding buffer F is used to promote the binding of DNA towards magnetic beads. And from here, 2 methods can be applied to separate the magnetic beads from the DNA, magnetic beads with DNA from the surrounding solution. The first method is magnetic rack, in which the magnet is located outside the tube. And the second method is magnetic rod, in which the magnet is dipped into the solution to transfer the magnetic beads. After securing the magnetic beads, unbound substance are washed off by wash buffer F1 and F2. And lastly, DNA is eluted from magnetic beads using TE buffer. For this kit, binding, washing, and elution steps can be carried out using automations. It is compatible with most of the automation instruments on the market. Some are using the magnetic racks, and some are using magnetic rods. We just need to program the protocol from the instruction manual to the instrument, and it will carry out the binding, washing, and elution automatically, and this can greatly improve the throughput. Let's look at the performance of this kit. It has been tested against different animal species using manual and automated extractions. Based on Table 1, we can see that DNA yield from swine, mouse, and human are higher than those from chicken, bovine, and elephant. This is related to the biomass content of feces sample. In terms of purity, all feces DNA has 260-80 near to 1.8 and 260-230 larger than 1, and this indicates high purity. Figure 1 shows that the extracted DNA is high in quality by showing intact band, no smearing, and no RNA contaminations. Figure 2 shows that the extracted DNA is ready for downstream application for 16S rRNA PCR. This kit is compared with our current product, FastDNA spin kit for Feces and kit from Tiangen, Omega, and Kaizen. Showing here is the result for swine feces extraction, but we have tested on other feces as well. They are showing similar trend and hence not displayed over here. And it is clearly shown that the new product has the highest DNA yield as compared to different kits. In terms of purity, all extraction kit has 260-80, quite near to the ideal value of 1.8. but only MagBeads has the highest value for 260-230, and this suggested that DNA extracted using this kit has the highest purity. In summary, the kit has shown its ability to extract DNA from different feces, and hence having wide applications. It also has high performance by able to extract high quality DNA of high yield and purity. Besides that, the MagBeads technology is easy to operate and possible to go for automation, and therefore allow high throughput and it is time saving. So before I end my presentation, let's recap on the workflow of microbiome research and how MPBio products can help in your microbiome research. The process of microbiome research is divided into 3 phases. The upstream involves sample collection and preparation, and it is widely accepted that mechanical lysis is the best method for environmental sample. And this can be supported by our fast spread instrument with range of adapter and lysing metrics. Once the sample is lysed, DNA extraction can be done using different kinds of extraction kits. And we have offered different extraction kit that is based on glass milk, spin column, and magnetic beads technology. The glass milk kit has yield advantage and suitable for sample with low biomass. Spin column kit is easy to operate and has overall good performance by able to extract DNA of high yield and quality. And lastly, magnetic beads is automation possible and suitable for anyone who look for high throughput. And all these extraction kits have trial kits that is free for request now, and let us know if you are interested to try it out. Lastly, the analysis is done once DNA is extracted from the sample. Analysis such as SPINeasy or shotgun sequencing are the common practice to generate useful information about the environment. With that, I would like to end my presentation today. I hope this presentation can give you a clearer idea on how to set up your microbiome research and to select the right tools to achieve your research objective. Thank you, everyone. And these are the references used in this presentation.
Okay. Okay, so what I'm going to go through here is a bit of the webinar giveaways. We'll be running 2 promotional activities for customers attending this webinar. So for customers from Southeast Asia, Hong Kong, and Taiwan, you do actually have access to free SPINeasy and MagBeads trial kits, about 5 preps. So what you're going to do is that you need to contact your local distributor. The promo code to use is 2020_MagSpin_SMP. So do quote this promo code to the distributor, and they'll reach out to me, and I can provide free samples for 5 preps for both the SPINeasy and the MagBeads trial kits. The second promotion activities is for Singapore and Vietnam only. So from now to December 2020, you actually get 5% off all fast prep consumables, both the lysing kits and kits. So the point of contact is Biolaboratories, who is our distributor for molecular bio in Singapore. Alright, with this, I'm going to leave you with my contact. So I'm Gariel Tay, the Regional Sales Manager. If there are any questions that you have for me or Ming, feel free to drop me an email or contact me at my mobile number. Alright, so as mentioned earlier, there's actually a post-webinar survey with 5 simple questions, out of which I think 3 is MCQ and just 1 open-ended. So link will be sent to your email. The link for today's slides will also be at the thank you note upon completion of the survey. Okay, thank you everybody for attending today's webinar. So if there's any questions, feel free to shoot to me. Thank you, and I'll see you again for our next webinar.